So the basic question is this: what proteins are in that tube, and what proteins are on that gel? Of course, anybody working with proteins has seen gels like this. This is an SDS page gel, and stained with a stain called Kumasi, which is a nonspecific stain that binds to all proteins. There's no way for you to really identify what each band corresponds to. All you can really say is get a rough molecular weight based on how these proteins are electrophoretically separated. Before we get into it, let's think about what exactly we want to do. We want to identify a protein, say a protein that comes out of a gel from a band that's stained with Kumasi. We don't know what it is. Well, first off, of course we do know that proteins are identifiable by their primary structure. Right? So proteins have a primary structure, which is their sequence of amino acids in a polypeptide chain. That sequence of amino acids stimulates folding in different ways, secondary structures, which then interact with one another to form tertiary structures, and then protein-protein interactions can stimulate quaternary structures of protein. And that structure tends to have function, and that is sort of an ID for a protein. But ultimately, it comes back to the primary structure, the sequence of amino acids from the N-terminus to the C-terminus of a polypeptide chain. And here's just a subset of the protein shown here in cartoon form. Of course, if we break that down into an atomic structure, we can see that each one of these polypeptides is really a chain of amino acids linked at a peptide bond, indicated with this blue line. And each one of those amino acids, of course, has a different R group. All right? So there's essentially a sequence that can be read off of this, and anybody working with proteins knows this very well. So you are familiar probably with Western blotting or techniques like this where we have antibodies raised to specific peptide sequences, peptide structures, secondary structures. That's one way of sort of isolating or identifying a protein in a mix of other proteins. But what we can also do is really just calculate the masses of the amino acids in that polypeptide chain, which here are shown the neutral masses for each one of the residues in this polypeptide chain. And each mass of each residue is different depending on the R group. If we sum all of the amino acids together and remove water, which would have been removed during dehydration synthesis, we get the mass of the peptide we're interested in. Okay, so now how do we apply this using mass spec? So protein identification by peptide mass fingerprinting. So say we have a large protein, say it's 50 kilodaltons, and we're interested in identifying what it is. We have no idea what it is. Well, the one strategy we can use, and probably the most common strategy, is to break that protein down. Now, you've heard a lot of different techniques of mass spectrometry. It's not absolutely necessary that we break it down. We could, if pure enough, ionize a full-length protein and measure its accurate mass. But of course, that's just the mass of a mix of amino acids that are in a polypeptide chain of some sequence order. We don't know. So what we do instead is we break down the protein using enzymes that cleave specifically at particular amino acid residues within that polypeptide chain. Trypsin is the most common. Trypsin cleaves C-terminal to arginine and lysine. It's a very efficient enzyme, very specific enzyme. So we can mix a protein with trypsin, which will cleave the protein into corresponding peptides. Each one of these peptides then has a unique mass based on its composition of amino acids. And that signature, if you will, is almost like a barcode for that protein. A protein of a given sequence will produce peptides of specific mass, and that list of masses can be used as a fingerprint, if you will, for that protein. That fingerprint, so if we, and we'll get to the mass spectrometry part of it here in a second, but those masses can be compared to databases that were generated by genome sequencing. So proteomics is heavily relies on genome sequencing. We can go to genome sequence databases and predict sequence of all proteins in a genome. We can, by prediction, we identify exactly where arginine lysine, all the residues are located in that protein. So we know exactly, we can predict exactly what peptide fragments should be generated through a theoretical or in silico digest of a sequence or protein. And that should produce a list of potential masses. So now the idea is that we break down the protein into its peptide fragments. We use mass spectrometry to accurately measure the mass of those peptide fragments, producing a mass spectrum. Okay, so this would be just an MS spectrum, so this would not be a tandem mass spectrum. This is just measuring whatever ionized peptides are in a sample. You would see a mass spectrum with M over Z in the X, relative intensity in the Y. This could be done primarily using either MALDI or electrospray for proteins. And once we have that, that's essentially the fingerprint for the protein, right? So if we cut a band out of a gel, and we'll talk about workflow in a second, we digest that protein or that band, we should get a list of peptide fragments of specific mass that should match some protein that has been theoretically digested from a genome sequence database. So we can search these numbers in silico. And of course, the more fragments that you match, the higher the probability is that you actually have identified a specific protein. Problem. You can see here, this is a perfect example. We have two peptide sequences. Imagine you're digesting all the proteins in the human proteome. You might run across two peptide sequences which have the exact same identical monoisotopic mass, but in fact are actually different sequences, suggesting that they're actually from different proteins. Right? So how do we resolve this problem? We can't fingerprint these because this mass can correspond to two different proteins. This is a problem that is compounded if you're interested in post-translational modifications, because of course now there's a whole host of additional masses that you have to add to your theoretical digest that correspond to modified residues on each one of the peptides, increasing the chances that this problem can arise. So, as Facundo clearly explained, we can use tandem mass spectrometry, right? We use the mass spectrometer as a mass filter, and we can look at fragments that are generated from peptides that we are analyzing. So imagine then for a second that we've taken a MALDI protein digest and we've analyzed it by MALDI. We've spotted. You hit it with the laser, you get ionization, you have now peptide ions in the gas phase, and you get a mass spectrum. And there's our peptide mass fingerprint. We can use that same mass spectrometer to isolate one of these ions. Okay, so now we've used the mass filter to kick out any ions that we're not interested in. We're specifically interested in a particular peptide ion. Once that's isolated, we can fragment it. Fragment that peptide into its corresponding fragments. Now, how does that work? So the basic concept and the most common mechanism for fragmentation for proteomics and peptides is collision-induced dissociation or collision-activated dissociation. So say we have this peptide FYTLHK, right? Peptide bonds indicated between each of these residues. We know, of course, each one of these residues is a specific mass. We isolate this peptide, and then we fragment and scan out the fragments to the detector. We may get fragmentation at the peptide bond between histidine and lysine, producing these two mass fragments. We may get peptides that are fragmented between leucine and histidine, so on and so forth, generating a list of fragments that can be matched to a database of a theoretical fragmentation of that specific sequence, right? The N-terminal fragments that are generated by this approach, CID or CAD, the N-terminal fragments are called B ions. The C-terminal fragments are called Y ions. And so what we would get from this type of experiment would be a peptide sequence. We've matched the exact mass of the full-length peptide sequence, and then we've observed various fragments throughout that peptide mass that have matched a peptide in a theoretical digest of a sequence database. And we can label it like so. We may not always observe all of the fragments. In fact, most times we don't. We see a fraction of those fragments. Again, same concept applies. The more fragments that you identify that match this theoretical peptide, the higher the probability it is that you've actually identified that specific peptide. So this you can do in a variety of different formats. Like I said, MALDI, electrospray are the most common. In MALDI, the convenient part is you can process a protein sample, break it down into its respective peptides, and then spot those onto a MALDI target plate. Let them crystallize with a matrix. Typically for proteins, we use alpha-cyano, synapinic acid, often for full-length proteins, also DHB. The list of matrices Facundo presented earlier. Those can sit on the shelf. They can actually sit on the shelf for a long time, and that's one of the advantages of using MALDI. So you can spot samples and then let them sit. Take them to the mass spectrometer, collect an MS spectrum. You could manually look at that MS spectrum and then say, oh, I'm interested in these 3 ions. I would like to sequence those 3 ions. You can then isolate them one at a time, generating a different tandem mass spectrum or MS-MS spectrum, and then take that information and search it in the database. You could do that manually. I did it a lot manually, actually, when I was in graduate school, because we didn't have quite the sophisticated software that we do now, which I'm going to talk about, data-dependent acquisition. So the instruments that we have now, the ABI 4700, is a great way to just do a simple protein identification out of a gel. That can be done in an automated fashion. So instead of you picking these 3 peaks, you can say, take an MS spectrum, fragment the top 6 most intense peaks out of that MS spectrum. And all of that data is collected very, very rapidly. So within 5 minutes, you've analyzed the spot, you've fragmented a number of peptides within that spot, and you now have both MS and MS-MS data, which can be searched in online databases that are actually free. So there's an online database, Mascot Search database, which is freely available online. You search it by Google. You can take mass spec data and search it directly, identify a protein that way. We can also do this by LCMS. You recall Facundo mentioned in electrospray, we often couple liquid chromatography directly to the mass spec. The advantage being that we are able to analyze many more proteins, many more peptides automatically without having to manually spot them onto a MALDI target. The other advantage is that we can get multiply charged states of bigger peptides, which might be outside the M over Z range of a MALDI, which typically we're looking between 800 and 4,000 M over Z, and we typically get singly charged peptides. In electrospray, we can get multiple positively charged peptides. So if we did an LCMS experiment, we might take a sample, and I'll talk about workflow in a second, separate by reverse phase liquid chromatography. UPLC is what we have coupled to the LTQ orbit trap, and that's actually running with a nano electrospray ion source. Analyze all the peptides that are coming off of the column in real time, meaning we collect an MS and very rapidly pick the top 6 peaks in that MS spectrum and do MS-MS on each one of those. And then take another MS, pick the top 6, fragment those. And you end up with half a gig of data within 75 minutes. That data can be searched very rapidly using software tools that we have. Proteome Discoverer is shown here, where I've taken yeast proteins that have been separated by reverse phase LC, the UPLC, and analyzed by LTQ Orbitrap. And you can see all of that data is searched in the theoretical fragmentation database to provide a list of peptides that were identified from the protein, each one of those with a corresponding MS-MS spectrum. Right? So here you can see an example. This peptide listed here vertically with all the possible Y and B ions. I sort of break down anything you're doing with protein mass spectrometry into two groups: the sample preparation side, which is a considerable amount of work, and the mass spectrometry side, which is also considerable, but typically for general protein ID is not going to change very much. Sample preparation, we have protein prep. You could be working with a protein extract. You might lyse your cells and have a whole proteome extract. You could have protein complexes, which I'll describe here in a second. You can pull down a complex, or you could have single proteins. Then you have to digest those. You could either digest in solution. That's possible, although in solution you have to keep in mind that if you are in a physiological buffer, your proteins are folded. The accessibility of cleavable residues within that protein might not be as high as they would be if they were denatured. And so in solution digestion, you have to take on with care, understanding that you may need to take more aggressive steps to digesting the proteins. Or in gel. So you could separate proteins by SDS page, in which case you've already denatured the proteins with SDS, separated them out onto a gel, and so they're in a perfect state for complete cleavage by trypsin or whatever enzyme you use. At the end of the day, whatever you do here, you can start with feces, you can start with, since we started on this track, I can't get it out of my head. Dirt. Yeah, you could start with, anything, right? As long as at the end of the day you end up with pure peptides in an organic solvent, typically acetonitrile, maybe methanol, and a little bit of acid, formic acid, trifluoroacetic acid. Talk to somebody before you take on this process about what would be the best solvent to be in for the type of mass spectrometry you're doing, because it's different for MALDI than it is for electrospray. And so if you end up in that state, chances are you're going to get great data back from your mass spec experiment. And on the mass spec side, you could either do an LC separation. I have here reverse phase liquid chromatography, which could be coupled to the Orbitrap. You might not separate it all and just do MALDI TOF. The beauty of separating is that you overcome a common problem with proteomics analyses, which is ion suppression. So even with one protein, going back to our MALDI concept, one protein digested into its corresponding peptide spotted onto a MALDI plate, you're likely only going to observe in the MS spectrum maybe 10 to 20%, depending on the size of the protein, of the peptides from that protein. Where are the other peptides? They should be there on the plate. You don't observe them. Turns out you end up seeing a lot of ion suppression of different peptide ions, depending on their sequence, how well they ionize. If you have one peptide that ionizes very efficiently, you end up actually not seeing things that don't ionize efficiently in comparison. So LC separates all those peptides and gives them, if you will, the best opportunity for ionization and detection, and therefore fragmentation, and therefore use and identification. Typical workflow. If you're working with proteins, you know about SDS page, you know about lysates. Let's start up here in the top left. I know there's a lot of arrows, but typical experiment might consist of lysate prep. You lyse some cells, maybe you do some fractionation, maybe you deplete, maybe you have a tag on a protein of interest, and you enrich, and that leaves you with some protein in a buffer of whatever choosing. You could take that protein and separate it by SDS page, as I described. And from that SDS page gel, and I would recommend precast gels for this, and we'll probably end up with some protocols online that will really help people get through some of these steps. Precast gels avoid certain contamination problems that we often see in protein mass spec. You can cut bands out, literally, just go with a razor blade and cut them out. You do have to do it carefully because you can have contamination issues. And you can take that little band, chop it into pieces, put it in a tube, and perform what we call in-gel digestion. You can dehydrate the gel piece, the protein stays in the gel, then you can add trypsin, which soaks into the gel, cleaves the protein and the peptides, then extract the peptides in an organic solvent that's ideal for doing mass spec. Alternatively, you could take that protein and do in-solution digestion. There are many different types of sample prep, and you may not fit into one category. It depends on how you're doing your experiment, how you're purifying your protein, if you are purifying it, if you're looking at many proteins that are not associated, like a proteome versus a protein complex. So you can digest in solution, get to the same result, where you have peptides that are generated from the enzyme in a tube. Then you need to enrich or clean up those peptides. That means removing salts, removing lipids in this case, removing, you would never want to have any SDS or any other polymers in a protein digest. And then take that and analyze it by mass spec. Okay, where things go wrong, since this is sort of a general discussion about protein ID. Watch out for contaminants in the sample preparation stage. If you're identifying proteins, that means anything that comes off of your body into a container that's holding your gel, or a tube that is then exposed to trypsin, becomes digested, and is now a part of your analysis. This happens more than anything with keratin, which can come right from your hair, skin sloughing off. It's a huge problem. You end up seeing lots of keratin and very little of what you're interested in. So that's something you have to watch out. We're interested in identifying what are all the components of that complex, right? So we pull down on one of those proteins. All the non-covalent interactions between the other subunits in that complex come along for the ride. So we affinity purify, and in this case, we did an experiment where we have a peptide epitope competitor or not, meaning we have a flag tag epitope that's not attached to anything in one of the samples, which should indicate any non-specific bands that should not be cut out for identification as associated with the complex. We run those on SDS page in gel digest, MALDI-TOF MS. This is actually done on an ABI 4700, just like the one that we have here in our facility, and then the protein's identified. So here's the SDS page gel. Here's the peptide block lane. This is stained with Kumasi. And then these are bands that are specific to the no peptide block or the protein complex lane. So we identify those, each one of those protein complex components, simply by cutting the band out, digesting it with trypsin, extracting the peptides, analyzing by MALDI-TOF. And so here are a couple of examples. This is a MALDI MS spectrum of this band right here, and you can see a number of peaks. Of course, those are just a small fraction of the total number of peaks we would expect if we had 100% detection of each peptide. And so we identify, actually in this experiment, we identified 2 new subunit components of this protein complex. Okay, I'm going to leave you just with some common questions that are very frequently asked, and then we'll move on to the quantitative aspect. How much protein do I need for identification by mass spec? Well, that is a question that depends on many factors, but just a rough estimate. If you can see it by silver staining, you can identify it by the protein by mass spec. So if you are working with proteins, you probably are used to running proteins on SDS page gels. You can stain those with different stains. Silver staining is probably one of the more sensitive. And so anything you can see by silver stain, you can typically identify by mass spec. The actual sensitivity of the mass spectrometer is better than silver stain. So even if you didn't see a band, you could cut out a piece of the gel and identify protein. So things that you don't necessarily see in that, on that gel through whatever stain you use, doesn't mean that there's nothing there. By mass spec standards, there's probably something throughout the entire lane. There are more compatible stains like Cypro Ruby and Kumasi Blue, which are better for mass spec out of gels. What is an appropriate volume for MS analysis? Somebody asked me this the other day. Well, if we were going onto a MALDI plate, for instance, we might have our purified peptides, and we would spot, say, 0.3 microliters of that. So think about that. If you ended up with a volume of purified peptides, and it was a mil, and you started with 5 nanograms of protein, you're not going to have much material in that 0.3 microliters. But that's really the volume we're at for a MALDI-TOF mass spectrometer. We'll go up to 5 microliters when we're injecting onto an LC system that goes directly into a mass spec. Can I identify proteins from unsequenced organisms, like a whale shark? This is also something that we can do. It's more difficult and actually requires you can't actually database search that type of information because there's no database that exists. So you have to do something called de novo sequencing, where you take a fragmentation spectrum and simply by the distance between the Y ions or the B ions, you can actually manually sequence the peptide that was fragmented. Of course, it requires that you have a good fragmentation spectrum. You could then take that sequence and do blast searches and try to identify corresponding proteins from other organisms that have been sequenced.